My name is David, and I'm a PhD student from University of Ostrava, where I study biology. And I will tell you something about how is Python code helping us to solve genetic codes in eukaryotes. At the beginning, I will introduce you eukaryotes, because you all probably saw in your life something like this, that life is divided into three main groups or the domains of life: bacteria, archaea, and eukaryotes. And you can see that archaea are somehow closer to eukaryotes, even in this textbook from 2005, and it's known for a long, long time. But nowadays, we know that eukaryotes are actually in group of archaea. And if we would zoom on eukaryotes, we would see a phylogenetic tree like this, which shows you how our eukaryotic organisms related to each other. And you all know animals, higher plants, and fungi. But you can see there's a lot of other things. But the reason why we don't know them so well is because they are usually unicellular. And majority, majority of them is unicellular, and the biggest diversity of eukaryotes is in these unicellular organisms. And they have different genomes, different shapes. And for example, I put some of them here. So this is Amoeba proteus from Amoebozoa. This is Trypanosoma brucei, which cause sleeping sickness. This is so-called brain-eating amoebae from this group, Heterolobosea. And it's called brain-eating amoebae because it can get to your brain through your nose, and then your chance to survive is less than 1%, I think. So there are some really interesting organisms you can see. And our lab is mainly focused on genomics and molecular biology of these unicellular eukaryotes, which are also called protists. And now I have to introduce you some basics about molecular biology. So you all know DNA. It consists from sugar phosphate bond and 4 bases: cytosine, guanine, adenine, and thymine. And cytosine is able to pair with guanine through 3 hydrogen bonds, and adenine makes pair with thymine with two hydrogen bonds. And RNA is very similar thing to DNA, but it's usually single-stranded, and instead of thymine, there is uracil, and there is also different sugar. There's no deoxyribose, but there is no deoxyribose, but just ribose. In case of proteins, it's a little bit more complicated because there are no just four bases. But there is at least 20 proteinogenic amino acids, which are bound together via peptide bonds. And those amino acids, they have different properties based on their side chains, which can be negatively charged, positively charged, uncharged, nonpolar, and many other things. And based on the primary structure, based on the amino acids in that primary protein, like the chain of the amino acids, proteins are folded to have different functions and shapes, and proteins can have structural functions, or they can function as enzymes. And basically, your body is made mainly by proteins. Also some fat, and it's not just this fat, but also lipid membranes on a cell, and also some genetic information in DNA and RNA. And what's the connection between DNA and proteins? All genetic information in our body is in DNA, and DNA, let's say, for proteins is like a plan how to make proteins. So DNA is transcribed into mRNA, and this is translated into amino acids. And there are two important things in this picture, and first of them is that always 3 nucleotides encodes 1 amino acid, and these 3 nucleotides, they are called codons. Oh, it's like King's speech. So this is first codon, second codon, third codon, fourth codon. And the second important thing here is that translation is terminated at stop codons, which are special codons, actually, which cause the stop of translation. And we know 3 of them, UAA, UAG, and UGA. And actually, this is so-called standard genetic code, and we all share this standard genetic code. But there are some exceptions from this standard genetic code, and they are called alternative genetic codes or non-canonical genetic codes. And actually, you're programmers, so you can think about this genetic code. It's just a basic dictionary, and if you want to translate DNA to proteins, or let's say mRNA into proteins, you just have this dictionary, and you always cut the mRNA into these framers and translate them according to this dictionary. Very, very simple. And as I said, majority of organisms use standard genetic code, but there are many organisms which are using alternative genetic codes. And it's nothing new. It's known for a long time, and usually in eukaryotes, and in our lab we are mainly focused on eukaryotes, usually there is UAR stop codons, which means UAA and UAG. They encode, not stop, but they encode some amino acid, and most often is glutamine, which is Q. And you can see that it's happening very, very often. In our lab, in last 2 years, we published 2 very important papers in this field. The first of them is about an unprecedented non-canonical nuclear genetic code with all 3 stop codons reassigned to some amino acid. And we found it in trypanosomatid, blastocystidia, and this is just amazing thing because all stop codons can code amino acid. So the question was how this organism can survive, and people found out that these organisms, those stop codons in these organisms, they have dual meaning. So if they are in mRNA not close to the end, they can encode amino acid, but if they are close to the end, they are used as a stop codons. And it's very amazing job, but it's not that amazing because I was not included in the authors. And, but the second project, this is just amazing because you can guess I was included in this one. So we discovered also 2 unprecedented nuclear genetic codes with UAG reassigned to encode amino acid, but UAA kept as a stop codon. And we found it in fornicate from this group, metamonada, which uses UAG to encode glutamine, and in a novel member of this group, Archaea, which uses UAG to encode leucine. And we don't even know how this organism looks because we never saw it, but we saw molecular data from this organism, and we found it as a contamination of this bug, Ligus asperus. And why is that so important? Because before, there was at least 13 coordinated reassignments of UAG and UAA in eukaryotes evidence before, and it suggested some evolutionary constraint. So it means when UAA stop codon translated, when UAA stop codon, when it encodes some amino acid, UAG always encode the same amino acid, or they both were kept as a stop codons. So people thought that there is some restriction that they always have to code the same thing, and we eventually broke this constraint, let's say, which is very important because things like this, they're helping us to better understand translation. And you can read more about it in our recent paper from last year, and I will tell you how we did that. And I will tell you Yotanema spiralis story, actually. So at the beginning, there was gecko. And gecko made something, And people from Charles University, they are very good with feces. And they isolated that anaerobic protist, and they are also very good in cultivation anaerobic protists. And at some point, they decided to sequence RNA from this organism. And we'll have a short break here, so I will explain you what you can sequence. You all heard that we are sequencing genomes. Yeah? But you can also sequence transcriptomes, which means that you are not sequencing this thing, but you can also sequence mRNA, which is sometimes more useful because in DNA there are non-coding things, and then it's hard to predict proteins. Because in these days, we know sequences of proteins thanks to sequencing mRNA or DNA, because it's very hard and expensive to sequence proteins, so it's way more cheaper, and it's cheaper and cheaper and cheaper to sequence DNA or mRNA. And when you sequence it, you obtain super short sequences, but then you can process those short sequences, and you can prepare something like this. You have a lot sequences like this one, and it always consists of two parts. This is called FASTA format, and there is a header, which starts with bigger than sign, and then on the second line, this is one line, there is actual sequence. And when you have this sequence, you can easily translate it into proteins if you know the genetic code. And you can use Python for this purpose. It's super simple. So they sequenced that, and then we took all those mRNAs and we searched in a huge UniProt Tremble database, which is database which stores information about proteins and genes and the genomes of a lot of organisms. And we were looking for similar proteins what we sequenced in other organisms. Because you, higher plants, also this protist, gecko, this organism, we all share a lot of genes. We basically share them, and they are very similar. So we check those databases, if there are some similar proteins, and we obtain some XML file. And this is not XML file, but I'm showing you this one because it's more human readable, let's say. And when you look for similar proteins to your proteins, you are doing it with one third-party program which is called BLAST, just amazing tool, but it's not important now. This is just output. And at the top, here is our sequence, let's say. The last line is the similar protein what we found in the database. And in the middle line, you can see how they are similar, let's say. If there is in middle line the same amino acid, it means they both have this position. They both have the same amino acid at the same position. If there is plus, the plus means that there is not exact amino acid, but there is some amino acid which has similar biochemical properties. And if there is nothing, it means that they are not similar in this position. And we were looking basically for sequences which had this asterisk in their sequences, because that means stop codons. And we found that a lot of sequences from this Eukaryotes spirale has stop codons in their proteins, and it would be not able to survive with standard genetic code. It would basically die because those proteins, they are important for surviving also. So what we did, we collected all those candidate sequences, and we performed something which is called multiple alignment. As I told you that we found similar proteins from different organisms, then you can, with some tools, you can put them with the same parts. You can align them. You can see that the same amino acid is aligned, yeah, in all of them. So this is from one organism, this is from second organism, third, and their sequences is not like shifted, yeah, it's nicely aligned. And thanks to this, we were able to check every position in those alignments where we saw those stop codons, And we were able to guess what amino acid is encoded by this stop codon. And you can see it also from this alignment that it's most probably that Q. You don't know most probably what Q means, and Q is glutamine. So from this, it's very obvious, but we did it like broad scale. We took like whole transcriptomic data. Whole transcriptomic data, it means that we took all mRNA sequences. So it was a lot of sequences. We performed the alignments, and some of them were just bad, so we discarded them. And then we collected some statistics, let's say. So we basically we basically checked every position in those alignments when there was UAG stop codon in our organism. We didn't care about UGA and UAA since we found that actually we performed the same, but I will not show you the data because there is nothing. They are normal stop codons. But only UAG stop codon was reassigned to encode glutamine. But when we checked positions with a 90%, like when we check positions where some amino acid was conserved in more than 90% of all organisms, or 50%, we saw that it is most probably glutamine, but it was only in 43% of positions, which is good, but not that much. Yeah? So we checked how canonical glutamine codons actually behave in this organism. And we found that the pattern is very similar to those positions from UAG stop codon. So we conclude that our organism is using UAG as a glutamine, while UAA is kept as a stop codon. And we checked that it kept UAA as a stop codon. And actually, why I'm here, yeah? Because now it's just about biology, right? And it's maybe not so enjoyable for you. So I'm here because almost everything was done in Python. And Python turned to be an amazing tool to solve these problems, because when you work with one sequence, you have one protein or one mRNA, you can, or one transcript, let's say, not mRNA, it's very easy. But sometimes you need to process like hundreds of thousands of those sequences, and you would be able to do that by clicking, but it would be super long PhD. Yeah. As I said, almost everything was done in Python, but I will not show you any actual code because I wrote the code 2 years ago, and it's very disgusting. Because in science, we are usually solving some problems and then we move on. So the code is very, very disgusting, and I don't even want to watch it. But I can tell you what packages I used. I used BioPython, which is just an amazing tool for working with sequence data, like proteins, nucleotides, and it's also great when you check those similar proteins in some huge databases. You can easily take the result of it, and you can parse it into Python in a very beautiful way. Then I also use pandas and matplotlib, but there is maybe no need to introduce you those packages. And actually, I was faster than I was supposed to be. I would like to thank all my collaborators, especially my supervisor Marek Eliáš. He's just genius guy, and my co-supervisor and great friend Tomáš Pánek. I would like to thank my financial supporters, University of Ostrava, Kačer, and Ostrava. I would also like to thank PyCon-CZ because I learned a lot from PyCon-CZ 2 years ago, a lot about pandas, matplotlib, and those things what I used. And I would also like to thank you for your attention.
Okay. Wow. Genetics. It's everywhere. But it is good that you can use Python and work with that. So, do we have any questions in person? Do we want to watch the code? I'm really interested in it because everybody knows that once you finish something, it's just obsolete and you need to work better and actually improve it. So
yeah, that's true. I can tell you one thing. I can tell you that the pipeline what I showed you, it took us like, I don't know, 1 or 2 weeks, and it's a long time to process one transcriptomic data. And now I started to work with some group of organisms, and they sent me like, I don't know, 75 genomes and 35 transcriptomes, and I didn't want to do it like months. Yeah? So I prepared better code, and that code you start it, and in 5 minutes you can in 5 minutes you have genetic code of your organism, and it's that code maybe is better, but it's yeah.
Show us.
Okay,
please.
But still it's very ugly.
I believe that there will be someone who could code review it and help you.
Yeah, I'm really afraid of it. Yeah. So okay, this one. Oh, guys, don't judge me too hard.
I wanted to ask, how much code was it?
How much code?
How much code?
If I would tell you,
to implement this work.
If I would tell you, you would take my job, because it's like these things. It's usually like 100 lines. It's super easy. Yeah, it's super easy for people who know how to code. For me, from the beginning, it was not that easy, but it's not a complicated thing. This is actually I've worked on some projects which were like way more difficult, but this is simple. And actually, this, yeah, when I will go fast enough. So.
Okay, great. Thanks for the question. We have another one.
Hi, is there something Python community could help you? Some technical problem you are struggling with?
Once more, please.
Is there a technical problem a Python community could help you? Some Python tools?
Actually, I think that I can solve everything with Python. And that's the beautiful thing about Python, yeah? I feel now, I learned how to code like 2 years ago, and I feel like a superhero. So
that's cool. I think that Peter wanted to ask you if the community about Python, so us, if we could help you with that. So
yeah. I didn't get the question properly, maybe.
No problem.
Actually, I want to make some of those tools. I want to make them open source, because I think that many labs can profit from them. I wrote some things also that you can very easily and in very fast way predict proteins from genomes, which there is many tools for that thing, but usually you need relatively big computational capacity, and also it takes time. And my script, it's super simple. It doesn't predict like all genes. It predicts only those genes what you are looking for, and it takes like 5 minutes to run. And the best thing, I added progress bar. It's just
cool. No, I'm serious.
It's very important progress bar, because then people know how long it will probably take.
You don't know if you will wait 5 minutes or 30, so it's important. We have another question.
Hi. I have a question about some previous code that, for example, have you seen? My question is pretty much, is there a lot of open source code connected to this that other, I don't know, aspiring bioengineers can use or can see?
I would say that all those packages what I showed, they are open source. BioPython, Pandas, Matplotlib.
My question is more that, for example, I myself, I learn by looking at some other people's code. So did you use as simple such a strategy? Just
code from other people?
Yes, about this subject, right?
Yeah, the problem is that when we work on something, it's usually a very particular problem. And science is usually about that you are doing something, but nobody did before. So usually it's not like that. Yeah, actually I took some code from our collaborators from Vancouver, I think, once, and modified and made way better code. But yeah, when I want to solve something, I'm usually just yeah, you know, Stack Overflow, but I'm usually checking just small pieces and then make something beautiful from it.
Awesome. We have another question.
Okay, maybe a little prosaic question. What was the main reason why you choose Python, for instance? You can choose another programming languages like MATLAB, for instance.
That's very nice question. Actually, I started with Perl, and then I tried Python, and it's so easy. It was so easy, and the learning curve from the beginning, it was like amazing. Now it's not that amazing, but still. Yeah? I was able to do things what people were doing basically for weeks or months. I just start some code, and I went for coffee. Because actually nobody in our lab, nobody was able to code. So I did like way better job in like few seconds than them like for one week. Yeah, so that's why I choose Python, because it's super easy to learn. And actually, biology now is full of Python. So also some very important and famous tools for genome assembly and such things, they are written in Python. And it's very good when you can check the actual code of some tools, and then you can add something or modify something. So, yeah.
That is true. And I think that Python will help with a lot of scientific projects in the future. And I hope that the community that we are building, that all of us here will be able to help you in this endeavor. So, do we have any further questions? Come on. I'm really glad that we have, okay, I'm coming. I'm really glad that we have this practical usage of Python.
Just one small thing about performance. Have you tried different Python interpreters before? Something like PyPy that helps a lot with speeding up some of your code, specifically with these functions. I've used it on DNA before.
No, I never tried. I don't know why. It was PyPy?
Yeah, just PyPy. I can't guarantee it's going to run by Python because not all libraries are supported, but if it does, it's going to speed your code like 20 times.
Wow, I will definitely check that. Thank you. Actually, my biggest improvement was when I implemented some multi-processing there, right? Not multi-processing. I was able to implement more CPUs for one job. Because I love Python so much, at the beginning I was not able to go for a Python workshop because it was very expensive. But then, luckily, one of my friends was bitten by a dog. So I went there instead of him. But still, I was thinking like it's not fair that it's expensive and no everybody can afford it. So I decided last year to create a workshop which would be for free for everybody, and it would be devoted to Python in bioinformatics. And last year there was people from 7 countries, and workshop was for free, and coffee breaks for free. And this year is even better. It'll be actually in 2 weeks, I think, or something like that. And everything will be for free. So lunches, coffee breaks, workshop, and people who will be there, they will also obtain some USB sticks and some nice things. And everything for free for everybody. This is my best achievement. Yeah, it's just amazing. And now it's full, guys. So, but for the next year there will be third year of this amazing workshop. So if you're interested in bioinformatics and Python, you can let me know, and I can sign you for next year. But please don't go there just because of lunches. Yeah?
This is awesome. We will talk about this a little bit later because we are doing something similar in France. So let's talk about that. So okay, thank you very much. And again, it was nice.